Hello, and good day everyone. We are from Group 1. In this video, we will be presenting out report on the cytology practical session. This is the list of names of our group members who have worked hard to prepare for this presentation. The contents of our presentation includes the staining technique, vaginal smear, organ impression, organ squashing, fine needle aspirate cytology, FNAC, urine smear, buccal swab, and the references used. The staining techniques that were used in this practical are the May-Grunwald-Giemsa, MGG stain and Papanicolaou stain.
These are the staining procedures for MGG stain. Due to the time limit, do pause the video if you need time to read through the procedures. Next, the procedures for Papanicolaou stain. These are the procedures for vaginal smears. Again, do pause to read if needed.
This is the microscopic observation of the vaginal smear. From the laboratory practical of vaginal smear using Pap stain, we can see that the slide is overstained compared to the reference which has a crystal violet staining. The superficial squamous cell has a dark pink appearance and the cell is in estrus stage. This can be compared to the reference, which also shows a estrus stage, nucleated epithelial cells, abundant enucleated cornified epithelial cells, granular cytoplasm and cells that are irregular in shape. The troubleshooting that can be done is to make sure that the staining time is accurate and appropriate.
These are the procedures for organ impression.
From the microscopic observation of heart impression, we can see some cardiomyocytes with nuclei that is shown by the arrow as compared to the reference photo. The staining that is used are the MGG stain under 40 times magnification. The microscopic observation of heart impression by using Pap stain under 40 times magnification shows that there is some macrophages infiltration and granulation tissue. As compared to the reference which shows lymphophagocytosis in pericardial fluid using H&E staining under 400 times magnification. The organ impression of normal lungs shows bronchial epithelial cells, alveolar macrophages, bronchial epithelial fragments consisting of ciliated columnar cells with terminal plates, benign metaplastic squamous cell, alveolar macrophages, and a large fragment of mesothelium with folding. From lung impression using MGG staining, we can observe the fragment of mesothelial cell sheet and alveolar macrophages under 40 times magnification. Compared to the reference which also shows fragment of a sheet of mesothelial cells and honeycomb appearance of dilated intercellular spaces using MGG stain under 200 times magnification. The lung impression using Papanicolaou stain, under 40 times magnification also shows the presence of mesothelial cells and alveolar macrophages. This can be compared with the reference that shows the structure of the mesothelium cells using Wright stain. The liver impression displays some clumps of well-differentiated hepatocytes by using Papanicolaou stain under 10 times magnification. While the reference photo using Papanicolaou stain shows hepatocytes that have abundant granular cytoplasm, round nucleus and a prominent nucleolus.
Next is organ impression on the kidney show present renal tubular epithelial cells. The nuclei is central and round with the nucleus is atypical, with granular cytoplasm. This picture is stained by using MGG stain under 40 times. This is impression on the spleen where normal cells show present of T cell and spleen endothelial cell. This picture is taken by using May-Grunwald-Giemsa stain under 40 times. In the picture with the arrow show present of segmented neutrophil, band neutrophils, lymphoid cell and erythroid precursors. The cells is stained by using Wright-Giemsa stain. For rat spleen, by using Pap stain and observe under 40 times, the cells show present of macrophage, T cell and B cell. To avoid a problem, make sure to set a timer for adequate fixation. The reference picture show present of lymphocytes, monocytes, macrophages and precursor cells using Wright-Giemsa stain. Next is organ squashing.
These are the procedures for organ squashing.
For rats heart squashing, it show myocyte cells with nucleus. The picture is taken by using MGG stain under 40 times. Reference also present of myocytes nucleus using H&E staining. For CAP staining under 10 times, the myocardial tissue with cardiomyocytes can be observed as compared to reference which the myocardial tissue with myocardial matrix can be seen using H&E staining. From the rat lung picture, the arrows show alveolar macrophage with few mesothelial cells. To obtain clear image of the cells, ensure to provide better staining technique. The reference picture is from the pig sample where fragment of a sheet of mesothelial cells and honeycomb appearance of dilated intercellular spaces can be seen. Microscopic observation under 40 times for CAP staining show mesothelial cells with abundant light blue cytoplasm and a round of nucleus found at the center. The reference picture is taken by using CAP staining under 200 times show reactive pleural effusion and normal mesothelial cells. Lung microscopic observation show mesothelial cells with abundant light blue cytoplasm and a round nucleus found at the center. To compare with human reference cells, present of pulmonary alveolar macrophage can be seen under 200 times CAP stain. From the liver organ squashing, for MGG staining, present of clumps of hepatocytes can be observed but without clear distinction between cells. Precaution when squash the organ is make sure to take small piece of tissue. The reference picture is taken by using right genes as stain. Hepatocytes have abundant granular cytoplasm, round nucleus and prominent nucleolus. Next is liver. From the cells, hepatocytes can be seen but with a very fine cytoplasm lining. The cells is observed under microscopic 40 times Pupaniculeo stain. The arrow in reference figure shows hepatocytes having abundant granular cytoplasm, round nucleus and prominent nucleolus. Move to kidney. The picture is taken under 40 times using MGG stain. The result observed from the tissue is present renal tubular epithelial cells. Central and round nuclei. Lack of distinct cell boundaries. Compared to reference, the picture is taken by using Sternheimer bin stain. The nucleus is atypical, with granular cytoplasm. CAP staining under 40 times for kidney show present renal tubular epithelial cells. The nucleus is atypical with granular cytoplasm. We can see from reference picture the proximal tubular cells, prominent of nucleoli and abundant granular cytoplasm. In the spleen, fragment of spleen endothelial cell, T-cell and monocytes can be seen under microscopic 40 times, May-Grunwald genes are stain. To get better result, improve the intensity of the staining. You can see from the reference picture, large lymphocytes characterized by multiple prominent nucleoli using right staining under 100 times. For CAP staining observed under 40 times, present of T-cell, B-cell and monocytes are seen. To improve, you can follow the time procedure to avoid over-decolorization. The reference is taken under 1000 times view using right genes as stain. The arrows show present of lymphocytes and precursor cells. Next is fine needle aspirate cytology.
These are the procedures for fine needle aspirate cytology, FNAC.
From our findings, the heart tissue shows cardiomyocytes with the presence of macrophages using MGG stain under 40 times magnification. While Utah Med 2020 shows macrophages infiltration at the heart with little collagenization using H&E stain. While in PAP stain, myocytes can be seen clearly with its nucleus under 40 magnification where it is corresponding to Sims et al. 2017 where the researcher found mononucleated cardiomyocytes using H&E stain. For liver, our lab finding is similar to reference from Jelisijevic et al. 2015 where clumps of hepatocytes with abundant pinkish cytoplasm and prominent nucleus can be seen. Our FNAC specimen was stained using MGG stain and seen under 40 times magnification while the reference uses Reit-Dienster stain. FNAC of kidney using MGG stain shows differentiation of proximal tubular cells but with poorly defined cell boundaries. But Pat et al. 2014 shows the presence of mature and immature plasma cells with reactive hepatocytes using the same stain. In PAP stain, our finding is quite similar with the reference where proximal tubular cells can be seen with centrally located and round nuclei. FNAC of the spleen with MGG stain shows presence of spleen fibroblast cells, T cells, B cells, monocytes and granulocytes where it can also be seen in the reference picture. In PAP stain also, T cells, B cells, monocytes and granulocytes can be seen, however, the staining technique need to be improved to avoid overstained. Next is cytology of urine smear.
These are the steps conducted in urine smear.
With Pap stain, our urine smear shows polygonal shape epithelial cells with large nucleus and transparent cytoplasm. While, in the reference picture, it also shows the presence of polygonal-shaped squamous cells with small pyknotic nucleus and abundant cytoplasm with the addition of superficial cells. For MGG stain, we only saw artifacts and no cells in the smear. Thus, to troubleshoot the problem, other staining technique need to be applied. Supposedly, we should see the presence of the cells like in Nickhausvat al. 2015. And lastly is buccal swab specimen. Buccal swab was collected using the specimen collection swab. The swab need to be removed carefully from the plastic wrapper. The swab tip need to be avoided from contamination by touching any surfaces. The swab need to be rubbed against the cheek in up and down motion for 20 times. The swab should be avoided from touching other regions such as teeth and lips. The swab was smeared along the microscope slide. After labeling, the slides will be allowed to air dry before staining with Pap stain or MGG stain. For MGG, after air dried, fix the smear in methanol for 5 to 10 minutes. From the buccal swab procedure, we are able to see the squamous epithelial cell of the cheek using MGG stain under 40 times magnification. The polygonal-shaped cell have centrally located and oval-shaped nuclei. Our findings is similar to the reference using H&E stain where normal squamous epithelial cell has polygonal shape with round or oval nuclei can be seen. These are the references that we used in our lab report. That's all from us. Thank you.